Hi everyone, I'm Ho Jong-yu from University of Illinois at Urbana-Champaign, and I'm in my second year of my PhD degree. I thought it would be nice to introduce my research as a brief presentation so that you could better understand and follow key points while looking at the poster. Hope this helps, and if you have any questions or suggestions about this research, please send me an email to this address written on the bottom of the slide. So today I would like to talk about the response of grass carp larvae to acoustic stimuli analyzed based on particle tracking velocimetry. So we'll get started. Grass carp has been one of the most notorious invasive species of aquaculture in the central United States due to their negative impact on the ecology and its fast spread. As a solution, different types of barriers and deterrents have been proposed to control their navigation. In this study, we focused on the acoustic signal among various barriers to influence the movement of grass carp, especially when they're at their early ages. Our main research objective is to analyze the response of grass carp larvae when they're exposed to sound. To achieve this goal, we would introduce a framework to quantify the response of carp larvae to acoustic signal via particle tracking velocimetry and define a relationship between frequency and the larval response to sound. So this video on the left shows you how our experiment was conducted. Approximately 200 larvae were placed inside the tank and they were exposed to a sound from a computer speaker installed on the near outside of the tank, which lasted for 1 second. Acoustic stimuli consisted of pure tones ranging from 100 to 1000 Hz, and the amplitude remained consistent at 60 decibel. The whole procedure of experiments were recorded with GoPro camera. To analyze our result, we applied particle tracking velocimetry algorithm presented on the right side of the slide. First, larval locations were detected from each image based on the pixel intensities. And second, their trajectories were tracked by quantifying the similarity of movement between two consecutive frames with cross-correlation method. The location of detected larvae were overlapped on the original image in this figure, which shows that majorities of detected larvae were captured by their heads and even closely located larvae were distinguished from each other. It also shows that the algorithm was successful to mainly capture larvae and neglect noises of the image, such as sediments and light reflections, as highlighted in this blue circle. Now, moving on to our result, what we first did was to analyze larvae response with bulk quantifications of their movement. We computed mean and 95 percentile velocities at each frame and plotted them as time series in these two plots. First time series was obtained from 300 Hz, which clearly shows sudden jump of velocities after the acoustic signal was introduced, while second time series below here, which was obtained from 400 Hz, doesn't show a clear difference between and after introducing the acoustic stimuli. These bar plots show how mean and 95 percentile velocities were changing in general for each frequency condition, and we can observe that for certain frequencies, the response of larvae to sound are quite vague, requiring additional robust quantification of larvae response to sound. So now we raise a question, is analysis based on bulk statistics sufficient to explain the response of larvae to acoustic stimuli? Which our answer would be of course no. As it was observed from previous 400 Hz condition, analysis is required. Further analysis is required to validate that sound can provoke larvae to change their movement. This figure on the right was obtained with identical 400 Hz experiment result, which also shows why analysis based on bulk behaviors are insufficient to represent general larval response to sound. First, standard deviation of mean velocities are too high, as shown in this first figure, which means that movements vary a lot by individual larva. The magnitude of non-dimensional velocities at each frame for individual larvae is represented with colors in these two figures, where red means high velocities and green means low velocities. While high velocities, red dots, are constant at are concentrated after 30th frame at this figure B, while in figure C, their distributions are random, which means that the sound did not have any impact on their behaviors. Even though larvae in figure B and figure C were exposed to identical sound, their behaviors vary as shown by different distribution of red dots, high velocities in these figures. Now we analyze the response of larvae based on their individual trajectories. We set up 4 conditions that needs to be satisfied for larvae to be considered as responding to acoustic stimuli, which are listed in the left side of this slide. Figure on the right shows time series plot of velocity, acceleration, along with their trajectory for a typical example of larvae that were identified as having response to sound. As you can see, peak acceleration and velocities are occurring at the 30th frame when the acoustic signal was introduced, with large displacement and trajectories as well. By counting the number of larvae that satisfied all 4 conditions and dividing that with total number of detected larvae at each experiment, we computed response ratio at each frequency. Sound of 700 Hertz was most effective to change larval movement, while clear relationship wasn't observed between frequency and response ratio, but rather consistent proportion of larvae were reacting to sound stimuli, with response ratio ranging from 20 to 30% regardless of the frequency condition. In addition, we analyzed whether there are any group behaviors observed as a response to sound. Figure on the right shows trajectories of every larvae detected in the image during entire experiment duration at 700 Hertz condition. We can observe that movement of each larvae was independent from one another, and sudden initiation of motion among neighbors did not startle nearby larvae to start their motions as well. Thus, we can conclude that sudden change in motion near the tone frame purely results from each larvae acknowledging the sound and their decision on how to react to the sound. We'll wrap up this presentation by pinpointing the most significant virtue of corresponding study is that larvae behaviors were successfully analyzed by applying particle tracking velocimetry, which suggests possibilities of various related future works. Thank you.